Hello, and welcome to this Pharma On podcast, part of our DMPK Insights podcast series. And my name is Scott Summerfield. I lead the metabolism group in our UK-based integrated metabolism department. And today I'm delighted to be joined by Dr. Dan Weston. Welcome, Dan.
Thanks, Scott. It's good to be here.
Just a short biography before we start. Dan's Ph.D. was in ion trap mass spectrometry, which he gained at Nottingham Trent University, and has held various positions, many of which have been associated with structural elucidation, including at Schering-Plough, AstraZeneca, and currently GSK, where he is scientific leader, providing cross-portfolio biotransformation support. Relevant to today's discussion, Dan's research interests include structural elucidation using novel mass spectrometric techniques, complementary analytical techniques such as ionization for data rationalization and insights into metabolite structure, in silico approaches for efficient data analysis, ambient ionization, and rapid sample screening technique. So in this DMPK Insights podcast, we'll be talking around a recent publication from Dan and GSK colleagues entitled A Discovery Biotransformation Strategy, Combining In Silico Tools with High-Resolution Mass Spectrometry and Software-Assisted Data Analysis for High-Throughput Metabolism. And this was published in 2022 in Xenobiotica. Dan, I know the author list includes several UK and US colleagues, so really global efforts by way of our strategy and publications. First off, be useful just to summarize kind of why studying biotransformations has become an important part of early drug design.
Sure. Yeah. Thanks, Scott. So I think it's fair to say we put a lot of effort into later phase biotransformation studies to support assets as they move from candidate nomination, into sort of early clinical settings and so on, and throughout that phase. But we have a chance to move into early discovery and really start to make an impact to design so that we can iron out a number of issues around metabolism, such as soft spots in terms of how we support clearance, and also to flag early signs of reactivity and, really get a chance to address issues around metabolite safety rather than having to mitigate them with a lead compound in later stages. So it's something that is really important to do, but of course then that needs a different kind of strategy. So we're not doing fast development, we're doing something that's fit for purpose and that meets the time scale for early discovery, particularly in LI and LO, where you have to meet more readily the demands of design, make, test.
And so just for the listeners, maybe just describe the key elements of the publication, the kind of strategy and what you went through to qualify the methodology.
Sure. So the publication really is a summation of the work we've done to really sort of begin with the end in mind, I guess. So it's really looking at what we did beforehand to support discovery in an ad hoc way, and then looking at how we might streamline that support to make it more relevant, to make it eventually scalable in terms of being able to meet higher compound numbers, but really looking at removing a lot of the pinch points where there's a lot of manual intervention or time where we're not actually answering the question, we're just being, let's say, inefficient with our time or in the way that we use that biotransformation expertise. We're looking to get to, a first set of rationalized data as quick as possible. So what we did was we looked at utilizing the skill and expertise within the team and the current instrumentation that we have. So this is based on the Thermo Q Exactive platform. So it's all Orbitrap based, using in silico prediction to really guide us not only in method choice, so minimizing method development using generic off the peg methods for UPLC separation and sample introduction, but then also to look at how we use our analytical capabilities and using in silico metabolite predictions to guide our DDA, data dependent acquisition, so that we can get relevant tandem mass spectrometric information to make structural calls. So it's really all about streamlining that process, making sure that we spend more time doing what's important, and then at the end that we get fit for purpose data that can drive decisions for med chem. And that's been fair to say, a challenge both culturally in terms of shortening that process, but still maintaining that quality. And we started with that in mind, so we really want to have that bulletproof analytics, and then you can sort of let the reins off to say, okay, this is now fit for purpose. We can do this a little quicker. Maybe we don't have to have the bar so high, but we wanted to start that way so we can always let it off. It's hard to build it in once you're running at speed. So that's really one of the things that we thought was important, and that's one thing that's sort of borne out in the paper. And that global alignment is something that is not only working well, it's something that we're really proud of, between sites, that data quality is so closely aligned. So it's been a work in progress to start small, get the basics right, and then move on to, and we just kept pushing it until it fell down, and that repeatedly, that's where we then needed to focus our attentions, as it were.
And so you use residual clearance samples, intrinsic clearance samples. And did you use precedented metabolic pathways to build the methodology?
Yeah, so we wanted it to be as relevant as possible, and it's something that I guess we've been talking a long time in the biotransformation community, really. I mean, within GSK, but also in the community about, how do you get the most relevant, let's say, qual type workflow it's referred to. So, you get a quantitative clearance measurement of, disappearance of parent, but also how do you get the most relevant look at metabolite profiles, or what's happening to drive that clearance where you've got disconnects, let's say. And we decided that being able to look at residual clearance samples made these data the most relevant. So these are samples that then we retrieve from the freezer in a given amount of time, so that we still have maximized integrity. And then we run these for the Met ID. So, you get that connectivity and relevance, you maximize that relevance, let's say, rather than doing an ad hoc incubation at 110 micromolar, let's say, and we're running right down at about 0.17 micromolar. So seeing metabolites of that single figure nanomolar, really challenging, but the data have been pretty amazing due to the sort of quality of the analytics, I would say. And this, to the second point, we use metabolite predictions to guide us. So that's what makes us able to use DDA to the best effect, looking in the right sort of chemical space to sort of continually guide that process. But that said, we are aware that we could be introducing a prediction bias there, and that's something that we're continually aware of. So the oversight of a skilled biotransformation scientist to look for unpredicted, non-predicted metabolites, however you would phrase it, is really critical. So this is software driven, but overall biotransformation expert led, if you like.
Yeah. No, so I know one of the key things that I recall from reading the paper is it's not really a top 3 or top 5 approach, of metabolites that you see in that incubation. So, the sort of intervention of the biotransformation experts still really important.
Absolutely. And it became one of the things that, I think we've been here a number of times within the community and, wider communities where software assistance, let's say, or software driven workflows are now, and quite rightly, part of what we do, because we no longer have the time luxury, particularly if we're looking to, come into the discovery arena and play in a meaningful way. So it's something that was non-negotiable for us, but really it's trying to shorten that time so that once we get first look at data rationalization, what we feel is there from predicteds, and then other ways of trying to find more relevant non-predicted metabolites, either manually or with additional software assistance, which is something that we're currently looking at. It makes that something that is a comprehensive look, but we've removed a lot of the manual scan by scan for the predictors. They're the easier to find, and they're the things that we want to reduce that manual intervention on, make it more joined up and make it more streamlined so that we can hit a tighter turnaround time, but still have quality data.
Yeah, and there's still the kind of opportunity to look for things like reactivity, reactive metabolites that may be there, even though they may not be the most abundant, on mass spec signal, but you'll still have intervention to see, unusual metabolites, etc., that might not come out in the standard sort of top 3, top 5 prediction.
Absolutely, and I think that's something that is touched on in the paper, but it's really something that we really do by default. That's rather than just doing Met ID, we provide the biotransformation part, so this is what's really relevant out of what we see, and how do we contextualize that to, what has the project asked, what is based on the structural moiety or that series, particularly what is relevant, so we would look then to bring in ways of contextualizing those data to make them the most relevant to the project, rather than looking at top X metabolites, let's say. We'd look at flagging a number of things that are important, but not just important based on intensity or caveated relative intensity, let's say.
Yeah. And I guess that's an interesting point, because obviously here the customer is medicinal chemistry. So, not only have you had to bring several processes together to take the white noise out to become efficient, but there's also technical and cultural and procedural changes that have probably taken place as well. So just thinking about that, what would you say the key learnings would be if you wanted this to be adopted by another group?
I think it's as much cultural as it is, sort of scientific and process driven, really, at least, in my experience and in our experience at GSK. One of the things that we started with was, as I mentioned before, you start with the end in mind. So what do you want? What does your customer need? And it's really been a way of trying to reach that both analytically as providing good data, but then being able to do something with those data at the end and still deliver in a meaningful timescale. So if it's with more than one team, or even a team split over a number of sites, I'd say that the alignment, this could be alignment between sites, between instruments, across a single team, whatever it will be, really, that alignment, so that it gives either site agnostic or user or instrument agnostic support, is really critical. And that's something that we made a big push on early on. It's really, really important. But I'd say one of the biggest things that we've always done and seen as important, but it's ever more critical here, is that dialogue between biotransformation, your DMPK project leader, and your end customer, so medicinal chemistry. It's really important, seeing as you now have, let's say, a 10-day window rather than a much wider window to deliver these data in. And we want to get that window continually smaller, to align even more relevantly with design, make, test. That dialogue and understanding early on before you start, so that you're fully aware of what the project needs are, not just, we have a disconnecting clearance, let's say, could you tell us what our top X metabolites are? There's a number of other things in there that are going on that aren't dictated by metabolite intensity, and being able to have that discussion ahead of time is really, really critical in ensuring that you get it right first time.
Have you seen any unexpected benefits, say, in the dialogue between Briggs, DMPK, MedChem, or other facets of the user approach?
I think the dialogue between biotransformation and DMPK to start with, with the wider sort of DMPK with our project leaders and between groups has been a real bonus for us. It's made that whole process tighter, much more well-defined and aligned between our US and UK teams for biotransformation, but also it's allowed us to be more mindful of how we reach out and position this solution, within DMPK, but also to our medicinal chemistry customers internally. But I think it also primes us then for, I think as we mentioned, this is our first iteration of this workflow. It's not how it's going to be. This is our first, our first iteration of this workflow that is the starting point. We're still aware that we have a number of gaps and we want to make turnaround time shorter, compound numbers higher. We build that in an iterative sense when the confidence is there and when the need is there, and then we move ahead. But it's one thing that's really cemented that need for making those communications as relevant and as often as you can, to really sort of understand what your customer needs, but so they also understand what you can or could provide, and how it's different to what biotransformation is when you're looking, clinical samples, looking at human safety and so on. It's a very different call, although you use similar techniques. And I think that's something that we've seen as a real benefit, because we've looked at this now at stripping away all the things that are not needed or not relevant in a discovery setting, but that sort of clarity of thought analytically, let's say, and the more data directed approach and an ability to get minimal method development, get it right first time, be more efficient. That's really had a knock on to the studies that we use to support development. So it's been a really useful process for the entire discovery development spectrum that we'd support. So it's something that I think was a surprise to us when we saw how really bulletproof analytics, and it's really the thing that we'd focus on, is getting that part right. The data could go into any software. The software that we detail in the paper is the ACD software, MetaSense, but it could be really into any vendor software, vendor agnostic software. It's really getting that part right and then moving into the software part once we know all that is as good as it could be.
Yeah, it's interesting to know that sort of kick into development has been useful. So I was wondering, because one criticism or one critique might be that, the heavy process automation dumbs down the role of the biotransformation scientist. So what would your counter argument be to that?
That's something that we've, I think, been aware of as a community for a long time. And when software was in its infancy, this is decade to decades ago, I guess I'm talking about, I was one of those people who worried about that. I think software has become, much more intelligent, much more useful and relevant to biotransformation. And it's really about allowing us to get rid of a lot of those non-value adding tasks. It's biotransformation scientists still have oversight of this workflow. It's really reducing the time to first set of rationalized data. But that really is where the software says, I have found these predicted metabolites, and it presents you with a list of data rationalized in terms of, the way that a human would, but it doesn't mean that oversight process is absent. Very far from it. It's needed there now more than ever, but I used to have that kind of worry myself, and I think a number of people had. But I think in terms of looking at where we're at now, in what we have to deliver and in what time, not just within discovery at GSK, of course, but as a community, the use of software, as long as it's pragmatic, and is always got the oversight of a skilled biotransformation scientist, I think is something that makes a lot of sense. Particularly if you're looking at reducing bias, it's something that we're looking at in the future now, so that you don't look at using your predictions first. You look at what the data are saying, and the data leads you, and then you can use predictions, manual intervention, a number of other, rules that a biotransformation analyst would use manually to find compound-related material. I think it's really about using these tools in concert. But that is, of course, really just to do the metabolite detection, metabolite characterization part. It really needs the skill of a biotransformation scientist and discussion with our end customer to give the biotransformation piece. What does it mean? What do these data mean? What value do they add? Rather than just saying, you have 9 metabolites and we think they look like this. It's really about that leverage of, okay, so what does that mean? What decision does it allow me to make? And that's something that firmly sits within our camp in dialogue with our customers.
You mentioned that the ongoing fact that this wasn't the first time, this wasn't the end product, that you continue to kind of iterate on the process. So that leads into thoughts about potential future developments. What would you call out as the important ones to look at?
Yeah, so it's an interesting one in terms of there is a need to apply this support rather to higher compound numbers as we look to scale this. And it's something that for our first look we used a GSK incumbent software. Made a lot of sense, of course, at the time. It was fit for purpose and it was integrated in a number of areas of GSK analytically. From the evaluation and from continual use and applying it sort of the coalface at and with projects, what we've seen is something I guess we've always been aware of, is that potential for prediction bias. So metabolite predictions are tremendously useful, of course, but it's the way that we apply them, and we've been looking at using manual intervention, manual data interrogation, to minimize that bias. But if we're looking at scaling and having a much higher number of compounds and or a shorter turnaround, we really need to get to that place quicker, where we're happier with the first look at our sort of derived data, our first look, and is really looking at bringing that down by using different data interrogation approaches and software. So it's something that we're looking at currently with the offering of mass metasite, and using that in a way that is much more data driven. The next thing there is looking at continuing that global alignment in terms of, once instrument time becomes the limiting factor, how do we build in redundancy, for time on the instrument? So if that is just based on, let's say, a single instrument per site, these instruments, of course, also have to support a number of other studies, development studies and so on, or ad hoc studies. So it's really about having alignment across sites with the right instrument types for redundancy once this becomes our limiting factor, which will be a nice place to be, of course. So, but that's where we are at the moment. So we've looked at continually aligning not only our approach, but our instrumentation that would support this in its future iterations, which is really important. And something that we touched on earlier is one of the unexpected benefits is that doing this now and not saying this is our final product, it always encourages us to look ahead for what's next. and it could be big changes, like new ways of looking at software, or it could just be the way that we run our samples and very small changes to that process, which is something that we do in a continual, let's say sort of, 6-monthly ongoing process. Okay, what worked well? What could we do next? We could save X hours to days just by doing this one thing, and there's a huge knock on. So it really has encouraged that as a mindset between teams, to really keep driving this so that our data can remain as relevant as possible to design, make, test, which is a pretty rapid paced process, of course.
Thanks. It's been a fascinating and insightful discussion, especially just trying to bear in mind that as scientists, curiosity and, challenging dogma is a really important part of, process improvement. So thank you. It's been great to have you. So cheers.
Absolutely. Thanks, Scott. It's been a pleasure. Thank you.
And to our listeners, thank you very much for tuning into this episode of Pharmaron's DMPK Insights series. We'd like to remind you that our DMPK webinar series is also available on demand, covering a variety of key questions related to DMPK science and drug discovery and development. So stay tuned for more podcasts in our DMPK Insights series. Thank you and bye for now.